announced after this public showcase. I am excited to introduce our top three teams: BPPC03, BPPC30, and BPPC13. Remember, the session is purely public showcase of the best research, poster, and life science innovations from this year competition. At first, I would like to call BPPC03. We will begin with the first finalist team, BPPC03, focusing on the crucial topic of reducing of carbon dioxide using bacteria. Team BPPC03, please start sharing your screen and begin your presentation. And remember one thing, you will have only 10 minutes. মুশফিকা জাহান, অন্তরা মাহির, মাহিরা চৌধুরী. Are you ready?
Yeah, we are BPPC 13. Will we start or BPPC 03 will start?
Oh, sorry. BPPC03, Fatematul Zahra Shaili, Fabia Bushra Rihanna, and Maisha Ryan Mumu.
Yes.
BPPC03, please share
Yes.
your poster.
Is our poster visible now? Hello, can you see our poster?
No.
Please wait a second. Is our poster visible now?
Yes, visible.
Okay, thank you. Okay. December is not feeling like December anymore, right? Why? Assalamualaikum, I'm Fatimata Zohra Shoili from Team BBPC 03. Today, our topic is reduction of carbon dioxide using bacteria. So, I have already asked a question that December is not feeling like December. Why is it not feeling like December? Because we are not feeling the actual winter. We are already in the mid of the December, but we are not feeling to wear a jacket or uh, while going outside. So, what is the reason behind it? The reason is the increase of global warming. And what is the reason of increase of global warming? That is rise of carbon dioxide. So we have come with a uh, we have come with a project that how to reduce the carbon dioxide from the environment. Here there are so many awareness programs where we are told to plant trees, but in our current days we can't plant trees in every area because of the sufficient of spaces. Here we have come with an alternative idea, that is we can use bacteria instead of trees, where trees can be planted. Like how can we use bacteria? Here we are proposing to use the bacteria named cyanobacteria, as it has minimal level of harmful sites or no harmful effects on environment. So how can we use cyanobacteria? We are proposing
Excuse me, your poster is not visible.
I'm sorry. Okay, I'm going to share it again. Is it visible now? Hello? Can you see the poster now?
Yes, yes, I see the poster.
Thank you. Sorry for the inconvenience. I'm again starting from where I was, that how to use cyanobacteria. We are proposing that we will cultivate cyanobacteria in lab under with the help of a BG11 medium, and then we are suggesting to dry it under the under the shadow of sunlight, or deep freezing, and then we will use that, and then we will grind it in powder, and we will use that powder in mixing it in water, or in mixing it with the cow dung, and it will form fertilizer form, and then we will apply this fertilizer on the soil of industrial areas where we cannot plant trees. When we will apply this fertilizer, like we are using the word fertilizer because it is a form, not because we are trying to grow plants. In the form of fertilizer, we will spread it in soil, and then after going to the soil, the cyanobacteria again will regain its metabolism, and it will start to reproduce by absorbing the carbon dioxide from the environment, and as a byproduct it can also produce oxygen from in the environment, which will be really helpful for us. So that was all from for the introduction and the method. Now I'm going to share my stage with my fellow mate, who is going to describe furthermore. Well, thank you for giving me the scope to present our poster in this uh, great stage. Hello, assalamu alaikum. I'm Maisha Ryan Momo from BBPCO3. I'm going to talk about the results of the entire project. Just like my fellow mate has said, that sorry, that why are we using cyanobacteria instead of plants? Well, cyanobacteria can go through the process of photosynthesis just like plants, and produce and intake carbon dioxide for their own to create their own food. They take up inorganic carbon, either be it dissolved in carbon dioxide or bicarbonate, and use it in as a carbon concentrating mechanism so that they can produce their own food and energy for them. And in this process, they can and they also release oxygen as a byproduct. What's more interesting is that why did the concept of fertilizer came is that they can actually also fix nitrogen in the soil. So if we use this product in on soils, most importantly the soils that are polluted or that are dry, it can also make it neutralize the soil, and will make it fertile and eligible for growing plants. And now, if you want to look at part of the main concept, that reduction of reducing carbon dioxide, well, we will be looking at an infographic here that is he, here. We can see that this thing actually shows some demographics from to an experiment where some cyanobacteria of different concentration were put into two cylinders of 1 meter height and another one of 10 meter height. We have seen that as the carbon dioxide level rises, the effect of carbon dioxide drop in column decreases, but when as the concentration increases, the drop in carbon dioxide increases too. So it we can understand that it also showcases weakness from us, that it cannot go to many far areas, but all at the same time it also gives us strength or an opportunity that it can be used in places like rooftops, which doesn't need much higher heights, and it can also be used in places like industries where there are not much space where we can actually build plants to put an effect like put enough effect for dropping the carbon dioxide. Now I'm going to share the straight stage to our another partner, Rihanna.
Assalamu alaikum everyone. Thank you so much for sharing me the screen. So far, my teammate has already described the situation and the thing, the project, what are we trying to do. Now, I will be present the SWOT analysis of our project in which I will be describing the strengths, weaknesses, opportunities, and threats of our project. Well, let's start with the strengths. Our biggest strength is that cyanobacteria are nature's silent barrier. They grow with just specific sunlight, a simple nutrient, and yet absorb carbon dioxide with surprising efficiency. Besides, the powder we produce from them becomes fertilizer, turning pollution into productivity. But even warriors have limits. Cyanobacteria are sometimes sensitive, that as one change in sunlight, temperature, or water quality can drop the intensity. Besides, huge areas cannot be covered easily, and large-scale growth needs carefully monitoring. Um, but however, it has significant opportunities as well, as this method can transform rooftops, farms, ponds, and urban spaces into living carbon dioxide capturing zones. Besides, it opens door for green industries, climate smart agriculture. What looks like a simple green water could become the future of natural carbon capture. Yet, threat remains. Contaminating microbes can invade, pollution can weaken the culture, and sudden climate shift, the very serious issue we are fighting against, can strike back and reduce their efficiency. But these issues can be solved in laboratories. So from this SWOT analysis, one thing is very clear, and we can conclude the whole thing that these tiny organisms cannot change the world alone, but with our support, they just might. Thank you so much.
Thank you, Team BPC03, for your excellent and insightful presentation on microbial solutions for carbon dioxide reduction. Next, I would like to invite Team BPPC30 for their showcase presentation on the fascinating area of AI-powered smart bee genomics. BPPC30, are you ready? Zoha Ahmed, Ramisa Fatima, and Ammar Fida, are you present? Am I audible, BPPC03 and 13?
Yes.
BPPC30, are you present? Zoha Ahmed, Ramisa Fatima, and Ammar Fida.
I'm sorry, can you just give me 5 minutes? My team members can come so that.
Tamanna, hello.
Yes, Apu.
BBPC 30 is with us. Maybe they need couple minute of time. So start with the Chittagong medical team if they are
Okay, Apu.
available. Okay.
Now we move to our team of this evening, BPPC 13, presenting their work on AI-based screening for early detection of breast cancer. Team BPPC 13, please begin your presentation, and your time is only 10 minutes.
Is our screen share visible?
Yes, visible.
I'm Sayeda Mushfika Jahan.
And I'm Mahira Chaudhuri.
We are first year medical student from Chittagong Medical College, Bangladesh. The topic for our poster presentation is AI-based screening for early detection of breast cancer. Breast cancer is the most commonly occurring cancer around the world. In 2022, it was responsible for 6,70,000 deaths all over the world. Late stage diagnosis and lack of awareness often leads to um, high mortality rate. Early diagnosis can be the most effective way of increasing survival rate. But our traditional screening methods that are available, like mammograph and ultrasound, have limitations like human error, less access to trained radiologists, and dense breast tissue problem. So, the integration of artificial intelligence in early detection of breast cancer can be the can lead to diagnosis accuracy. So, the objective of our study is to find out the applications and benefits of using artificial intelligence based screening in early detection of breast cancer. Now, the method by which artificial intelligence detect breast cancer from thousands of mammographs will be described by my teammate Mahira.
So the AI takes input in the form of digital 2D mammogram or digital breast tomosynthesis in 3D format. The input is then pre-processed, as in the image is enhanced, increasing its clarity, reducing artifacts, and denoising it to prepare it for better detection. The AI model then analyzes the mammograms, checks for malignant versus benign tissue, checks for subtle patterns, and detects the lesion. The AI then classifies the detection according to malignancy by analyzing its huge, vast data set, and then ranks the malignancy according to its own scoring model.
Now, the key findings of our results study are AI reduces the rate of interval cancer by 30%. It flagged 76% of mammograph that had been linked to interval cancer. Then it flagged 90% of misreading error where the cancer has been visible on the mammograph but misinterpreted by the radiologist. It also reduces false positive by 37.3% and biopsy requests by 27.8%. Now we have two graphs here. At first, the graph on the left side is a comparison graph between when the mammograph is read by radiologist alone and when it was AI assisted. So from the graph, we can see that when AI was used in reading the mammographs, it increases the rate of positive report, it decreases the false negative report, and AI acts best when used as a second reader. Now, the graph on the left side is a receiver operating characteristics graph, and in the graph on the y-axis it shows sensitivity, that is the rate of positive reports, and on the y-axis it shows specificity, that is the rate of false negative report. So the higher is the curve, the better is the model. So from our studies we found out that when AI is used in reading mammographs, it shows better result. Now, from our overall study, we can say that AI detects breast cancer early. It can be very convenient in rural areas where there is less access to trained radiologists. Also, it can reduce the workload of radiologists. But AI has some limitations like algorithmic bias, data privacy and security, and large volume of data dependency. So the challenges of adopting AI screening are, at first we have to break the black box nature of AI so that it become more reliable to radiologists. Then ensuring clinical validations and ethical and legal guidelines are also must. So in conclusion, we can say that AI-driven screening ensure earlier detection of breast cancer, reduce diagnosis error, and it can help radiologists greatly. So thank you everyone for your time. Our presentation is here.
Thank you, team BBPC 13. Now we move to our final team of this evening. Team BPPC 30, Zoha Ahmed, Ramisa Fatima, and Ammara Fida. Team BPPC 30 for their showcase presentation on the fascinating area of AI-powered smart bee genomics. Team BPPC 30, please share your poster. Team BPPC 30, are you present?
Tamanna, maybe Zoha is with us. Hello.
Yes, Apu.
Maybe Zoha is with us. Hello, Zoha.
I think she can
Hello.
experience with some technical problem.
Tamanna, call again the team.
Okay. Team BPPC 30, are you ready?
Am I audible?
Yes, audible.
Okay. So, Assalamu alaikum and a very warm welcome to the respected judges. My name is Amara and I'm presenting this work along with my group members Ramisha and Zoha. We are from the Department of Computer Science, ComScience University, Islamabad. And our poster is titled AI Powered Smart Bee Genomics to Predict Honeybee Colony Collapse. The first question which comes in our mind
Excuse
after
me,
hearing
Amara, please share your poster.
okay. Uh, Zoha,
I'm
can you?
sharing the I'm sharing the poster on the screen. Is it not visible?
Yes, it is
Yeah,
visible.
Okay.
okay. So our poster is titled AI Powered Smart Bee Genomics to Predict Honeybee Colony Collapse. So the first question which comes in our mind that why we have chosen this topic, as we all know that honeybees are responsible for pollinating nearly one third of global crops, but their populations are declining rapidly because of diseases, parasites, pesticides, and climate stress. Um, Traditional beekeeping methods rely on manual inspection, which is slow, it's inefficient, it's inconsistent, and it often detects problem when it's already too late. So we asked a question: can technology help us detect bee health problems earlier and prevent colony collapse? This is the main idea which motivated us to present a poster on this topic. Our concept is basically it is inspired by a real research study which was published in Scientific Report by Carline and his colleagues in UK. The study sequenced the gut microbiome of bees from 23 hives and used AI models like different AI models like logistic regression to predict which colonies would survive this winter. They found that microbial composition could predict colony collapse, and this proves that genomics and AI can reveal early warning signals. Or posted this scientific foundation and proposes a complete smart AI system for beekeepers. Now, my teammate Zoha will explain you further that how basically this whole system will work.
Assalamu alaikum. My name is Zoha Ahmed. First of all, I'm sorry for the delay. There was a technical error. Also, my teammate Ramisha couldn't come because of some emergency. I hope you will understand. So, I will explain how our model will work. The smart bee genomics will follow the methodology. The first step is the sample collection of the bee DNA along with the hive debris, pollens, and gut microbiome samples. These materials will provide information about both the bees and the pathogens or microbes affecting the colony. In the second step, we will do the sequencing of that genomic data. The genomic sequencing, which will detect early molecular signals such as stress related genes, harmful mutations, pathogens, and changes in the hive microbiome. The third step is the most crucial one as it involves the use of AI. So basically, we will use our machine learning models that will extract hidden patterns from the genomic data, including disease signatures, levels of genetic diversity, and climate related stress markers. After this, our AI models will estimate the colony collapse risk. Okay, so they will predict if there is going to be any disease outbreak and distinguish weak colonies from the strong ones, and they may even forecast honey production potential of the colony. So yes, and the final step will be the visualization of the output, in which the system will generate a hive health dashboard, and which will it will decide that what steps are needed to be taken in for the colony. So this is our conceptual model. It does not have any existence yet, but we have shown some mock scientific results that if we follow with this model, we are confident that our AI model can achieve 92% accuracy in identifying early signs of colony collapse. Even the harmful pathogens can be detected up to 3 weeks earlier the appearance of the symptoms, allowing timely intervention. Um, yes, that's all. That's how our whole system will work. And if we To conclude this, our smart bee genomics framework will demonstrate how integrating metagenomic sequencing with artificial intelligence can significantly improve early detection of honeybee colony collapse. While traditional genomic tools identify the pathogens or microbes present in a hive, the AI layer which we are adding will allow it to use the ability to interpret complex patterns and combine multiple stress indicators and generate accurate predictions about colony health. Thank you so much. That's all from our side.
Thank you, Team BPPC 30, for presenting your work so clearly. The integration of AI and genomics to address colony collapse is highly compelling. Thank you to all our participants for their hard work and excellent presentations today. Each poster was unique and showed great effort and creativity. Now, the moment we have all been waiting for the winner of the poster presentation. Now, I would like to announce firstly, second runner-up. The second runner-up BioBangla International Poster Competition 2025 is BPPC 30, Zoha Ahmed, Ramisa Fatima, and Ammar Rafida. Congratulations, team BPPC 30.
Thank you so much, ma'am.
Thank Congratulations. And the first runner-up BPPC 03, Fatimatu Zahra Shaili, Fabiha Bushra Riana, and Maisha Ryan Momo. Congratulations, team BPPC 03. And the winner of the BioBangla International Poster Competition 2025 is BPPC 13. Congratulations, Saida Mushfika Zahan, Antara Maher, and Mahira Chaudhuri. Thank you everyone for joining. Congratulations to the winner and the runners-up. Appreciation to the all participants. With this, we officially end today's BioBangla Poster Competition 2025 and have a great day.